So hello students and welcome back to another video tutorial by Genome Biotech. In this video tutorial, we would be talking about the process of transcription as well as looking at the process through theory of Pathfinder as well as through the animated series of the book Watson. So what is transcription actually? Transcription involves the synthesis of an RNA chain. When you are going to biosynthesize an RNA chain over the DNA, then the process is commonly referred to as the transcription. So transcription is a process in which the RNA chain is going to be biosynthesized over the DNA. Then it is referred to as the process of transcription. Okay. Now, how does this transcription takes place? There are two strands over the DNA molecule which are present, and those two strands are the one which among which one strand will be used as a template strand for the biosynthesis of the DNA of the RNA molecule with the help of the enzyme that is referred to as the RNA polymerase. So the DNA will act as a template strand, okay, antisense strand. You can look here. The transcript is identical in sequence with one strand of the DNA. The transcript which is identical to the sequence is called as the coding strand, and the complementary strand over which the transcript is being biosynthesized is commonly referred to as the template strand. So there are two strands of the DNA molecule. I'm just making the two strands in front of you. Please have a look. Now see, this is a DNA molecule that you can find. If this is a DNA molecule that is present in front of you, there are two strands over the DNA molecule. Okay, 5 prime, 3 prime. This is a strand over the DNA molecule. Now this strand which is present over the DNA molecule has to be transcribed. This DNA molecule has to be copied. This DNA molecule will be copied with the help of an RNA polymerase in order to form the mRNA molecule, in order to form hnRNA or mRNA molecule. So it will produce an hnRNA or an mRNA molecule. This is the hnRNA molecule which is being produced. I'm calling it as heteronuclear RNA because it will have both introns and exons present within itself. The introns and exons will undergo splicing in order to form mRNA. It will have introns plus exons present within itself. When introns and exons are present within itself, these introns and exons will undergo splicing. When they will undergo splicing, the RNA that would be produced will be referred to as an mRNA molecule. So this strand of the DNA which is being copied is referred to as the template strand. Template strand. Please do make it along with me. Template strand, and the other strand is referred to as the coding strand. The other strand complementary to this template strand will be referred to as the coding strand. I'm also making the other strand in the form of a dotted line. This strand will be referred to as the coding strand. Now this is a misnomer. Okay, this is a misnomer. The coding strand is actually non-coding. The template strand is actually coding. This name is coding strand, but this strand is not coding. Okay, so transcription unit, the segment of the DNA which is involved in the process of transcription is referred to as transcription unit. Okay, so the part of the DNA that is involved in the process of transcription is called as the transcription unit. So how this transcription unit is present, that we have to see. The segment of the DNA that would be involved in the process of transcription is referred to as the transcription unit. In eukaryotes, a transcription unit usually carries the information just about a single gene, whereas in others, it carries the information about multiple number of genes present. Okay. So eukaryotes are monocystronic and the prokaryotes are polycystronic in nature. In eukaryote, a transcription unit typically carries information of just one gene, and so it is referred to as monocystronic. Therefore, codes for a single RNA molecule or a single protein in bacteria, a set of adjacent genes are often transcribed in the form of a single RNA molecule, and that is referred to as the polycistronic mRNA. There are two terms which are being used: monocistronic, and another term it would be given here, not given, polycistronic. Polycistronic. In the case of polycistronic, you just have a single promoter region and then you have several number of genes. There is a single promoter. Supposingly, there is a promoter which is present here. This is the promoter. And there are three genes: Z, Y, and A. Z codes for beta galactosidase, Y codes for Z, Y, and A. Beta galactosidase, permease, and transacetylase. These are the three genes which are present. After the promoter, these three genes are present. So the three genes will be transcribed by the same promoter and will produce the same type of a transcript. This would be the mRNA transcript that would be produced. Okay, this is the mRNA transcript, and this mRNA transcript will produce three different types of protein: first, second, and third. So three different types of proteins can be produced. This is referred to as polycistronic, single promoter producing the transcription of more than one genes. In the case of monocistronic, which occurs in the case of eukaryotes, polycistronic, the type of arrangement occurs in the case of prokaryotes, and in the case of monocistronic, you have a single promoter sequence that is present. Along with the single promoter sequence, you just have a single structural gene. Now, supposingly there is an hexokinase gene that is present in the case of eukaryotes, hexokinase gene. So, hexokinase gene will only have a single promoter. So, this is the hexokinase gene promoter. Okay, this is hexokinase gene promoter. A single promoter is present. A single promoter is present, and that is referred to as monocistronic in nature. Okay, another one is said to be polycistronic. One is monocistronic in nature, and another one is said to be polycistronic. In eukaryotes, a transcription unit carries the information of just one gene and therefore codes for either a single RNA molecule. In the case of bacteria, adjacent genes are often transcribed in the form of a single transcription unit. Anyone has any doubt or problem, you just ask me. And the immediate product of the transcription which is being produced is commonly referred to as a primary transcript. Okay, the transcription unit can be simple or they can be complex transcription unit. Primary transcript produces from single transcription unit is processed to give a single type of mRNA. There is a single type of mRNA being produced from the primary transcription unit. Whereas in the case of complex transcription unit, which are quite common in multicellular organisms, the primary RNA can be processed in more than one way, leading to more than one type of an mRNA being produced. Now, what happens in the case of a complex transcription unit? The complex transcription unit can be processed in more than one manner. It is not polycistronic, it is monocistronic, but the transcription can proceed in more than one way, leading to the formation of more than one type of an mRNA molecule. There are several different types of mRNA molecules that could be produced because Naya, the diversity of the mRNA being produced is due to diversity in case of mRNA being produced. Diversity in case of mRNA. In case of mRNA production. Diversity in case of mRNA production in the case of eukaryote. In eukaryotes can be because of reading frame shift, a, a phenomena which is referred to as reading frame shift, alternate splicing. I will describe all these things in my later lectures. Alternate splicing. Third is trans splicing. Splicing. Or it could be RNA editing. RNA editing. These are the different processes that could be used in order to generate the different diversity of the mRNA molecule. Okay, and such type of transcription unit which undergoes such type of modification in order to form more than one mRNA is referred to as the complex transcription unit. They are referred to as complex transcription units. Okay, these are the complex transcription unit which are present. Now, from this point, RNA polymerase moves down the along the template, synthesizes RNA until it reaches the terminator sequences. Sequences prior to the start point are referred to as upstreams, and sequences after the start point are referred to as downstream sequences. So there are two different types of sequences. Sequences have been named as upstream and downstream on the basis of where they are present. Just before the start point is referred to as upstream, another one is referred to as downstream. So I'm just taking the animation and showing it to you. The two things that must be shown to you. Now, this is the double stranded DNA molecule. Among this, the double stranded DNA molecule, when you are going to listen to the animation, then I
To
have
an
already
RNA
told
strand.
you that this is the RNA strand which is being biosynthesized. Now, out of the two strands of the DNA shown here, only one will be copied, while the other will behave just as a strand. So out of these two DNA strands, if I'm going to make the two DNA strands, just 1 minute, I'm choosing the colors so that it could be visible to you. Okay, I have taken a DNA strand. Now, over this DNA strand, if I'm going to attach an RNA polymerase, supposingly I have attached an RNA polymerase, and this RNA polymerase is moving over the black strand. And then there is another strand of the DNA that I'm going to show with red color in order to differentiate the two strands from each other. Okay. Now, supposingly, this RNA polymerase is going to move in 5 prime to 3 prime direction. It always moves in that direction. So if it is moving in 5 prime to 3 prime direction, okay, 5 prime to 3 prime direction, then which strand will be called what? Please indicate it with the options provided. Please indicate it in the chat box that which strand will be called as what, which would be the template strand and which would be the coding strand. You look at the black strand. The black strand is here called as, tell me now, try to answer. This is referred to as the template strand. Template strand. This strand, which is called as the template strand, is also referred to as misnomingly, also referred to as non-coding strand. Non-coding strand. Because the transcript which would be produced, because the transcript that would be produced would be exactly same towards for this is the transcript produce. Okay, because the transcript produce will be exactly same to the other DNA strand, the other has been considered to be as a coding strand. The red one is called as coding strand. Coding strand. Okay, so kindly do remember by the term template strand rather than coding strand. This is a misnomer and it could cause your questions to get wrong. Okay, so this is referred to as a template strand. The other one is referred to as coding strand. Any doubt, any problem, you just ask me. Okay, now moving forward, this is how the process of transcription will begin, clearing all the drawings and moving forward. Any doubt, you can ask me. If you feel any doubt, you just ask me. Is this clear in your mind? Now, supposingly, clear. Now, supposingly, this DNA molecule has several different types of genes. Now, first gene is present here, another gene is present here. Then, such type of mRNA that would be produced, or such type of arrangement of genes is referred to as polycistronic mRNA molecule. Polycistronic cistronic mRNA, oh sorry, polycistronic mRNA molecules. This is polycistronic mRNA molecule that is going to be produced. Such type of polycistronic mRNA molecule occurs in the case of E. coli. That is in the case of prokaryotes. That is, it takes place in the case of prokaryotes. Example, it is the one that is involved in the formation, in the transcription of lacoperon, arabinose, arabinose operon, lambda phage, lambda phage operon. So these are some of the examples that should be learned, and they are all the examples of such type of conditions that are going to be operational. Okay. This is referred to as polycistronic mRNA molecule. Such type of polycistronic mRNA molecule are going to occur only in the case of prokaryotes. In the case of eukaryotes, the above is not true. In the case of eukaryote, there is a uh, promoter, and that is encoding for the formation of a single gene product, that is referred to as monocistronic type of transcription. Okay, so we must move now forward and look at it. Now, deleting all the things from here, and let us have a look at the animation also.
The primary enzyme of transcription is RNA polymerase.
Okay, so this is referred to as the RNA polymerase that is going to move forward. Now, all the sequences beyond the start point of transcription, now supposingly there is a start point of transcription, and I'm just marking the start point with a blue color. Now, supposingly this is the start point of transcription. From there, the transcription has started. Okay, this be the start point of transcription. Start point of transcription. Start point of transcription. Point of transcription. So all the sequences after the start point of transcription are referred to as downstream sequences. Such type of sequences beyond this start point is referred to as downstream sequences. Downstream sequences. All the sequences prior to the start point, all the sequences before the start point are referred to as upstream sequences. These sequences are upstream sequences. Those after the start point, downstream. Those before the start point are referred to as upstream. Downstream sequences are always written with -10, -35. Such type of sequences are referred to as downstream. Upstream sequences are always written as +10, +20, and so on. This +10 indicate that it is 10 base pair before the start point of transcription. -10 indicates that it is 10 base pair down the start point of transcription in the direction of transcription is taking place. Now, when you compare one gene with another, now supposingly there is a very long sequence that I'm taking. Now, this is a confusion state that arises in the minds of the student. Supposingly, this is an RNA polymerase that is working on one gene. Another RNA polymerase is working on another gene. Okay, this gene would terminate here. This is the next gene that is being transcribed, and the process of transcription is taking place. Now, what is upstream and what is downstream? The term upstream and downstream can only be taken for the region of the DNA that is undergoing transcription. Okay, this is very important. Please do note it down. The term upstream or downstream can only be can only be used for the segment of the DNA molecule undergoing transcription. Okay, so both the transcripts will be labeled separately. Now, how they would be labeled, that we have to see. Please look here. See, this is what I had to tell to you, that this here in this particular region, if this is a gene, from here till here, if this is a gene, I'm just taking another color for this particular gene and another scale. If this is a gene that is going to be transcribed, okay, then all the sequences before this gene, all such sequences and all such sequences, they will be referred to as, with respect to this gene, this side it is referred to as downstream. This side it is referred to as downstream. This side it is referred to as upstream. Now for the other gene that I am going to take with another color, supposingly red. Okay, now I've taken another color that is red. And if this is going to be red, this is another gene that has been started, and here it has been, I'm assuming that it is going to be terminated here, it has been started here. Then all the sequences before this point will be called as downstream. All the sequences after this point will be all the sequences after this point will be referred to as downstream. All the sequences before this will be referred to as upstream. So those sequences which could be downstream for a particular gene are actually upstream to its preceding gene. वो सारे सीक्वेंसेस जो डाउनस्ट्रीम हो सकते हैं एक जीन के लिए, दूसरे के लिए अपस्ट्रीम होंगे. So what I am trying to state here is that those sequences, thus we can conclude that that those sequences which acts as downstream for the one gene could be acting as upstream for the other gene, for the other gene. So this is the major difference between them. So one sequences which are downstream for one gene could act as upstream for the other gene. So this is how upstream and downstream has to be regulated. Any doubt, any problem, you just ask me. Is this clear in your mind? Is this clear in your mind? Okay. So now moving forward. Now moving back to the Pathfinder, I have already told you, explained you everything about this particular point. Deleting all the annotations and moving back to Pathfinder. Just a second. So Pathfinder is visible to you now. Okay. Now, polymerase transcription. We know that the RNA polymerase that is present is the major enzyme in catalyzing the process of transcription. This RNA polymerase is consisting of the following subunits: alpha, alpha dash, beta, beta dash, sigma, as well as the omega. So you can find that this is the subunits which are present. Alpha 2, beta, beta dash, and the sigma factor could be present, can be synthesized into two different components. The components which are through to which these can be biosynthesized are the core enzyme and the sigma factor. Now, the RNA polymerase can be categorized into the following two different types: the core enzyme and the sigma factor. The sigma factor is the one that is going to screen the mRNA molecule in order to initiate to find the regions from where the initiation is going to be taking place, in order to find the region where the transcription is going to be initiated. The sigma factors, the sigma subunit is required for the assembly of the core enzyme and plays an important role in promoter recognition. Alpha subunit plays a role in interaction with the RNA polymerase. It is alpha subunit that is going to play a role with interaction with RNA polymerase. Beta and beta dash subunits are the other subunits which are making the catalytic site or catalytic center of the RNA polymerase molecule. The catalytic activity of E. coli RNA polymerase is provided by a heterotetramer composed of beta and beta subunit and two copies of the alpha subunit. Now, there are two copies of the alpha subunits which are present. There are two copies of the beta subunit which are also present. And these copies of the alpha and beta subunit are going to make the structure of the RNA polymerase enzyme. They will togetherly constitute the structure of the RNA polymerase. Now, how they are going to make the structure of the RNA polymerase, that we will have to see here. So please do have a look here.
When you have completed this exercise, you should understand the structure of RNA polymerase, understand the phases of the transcription cycle, and understand the differences between transcription and replication.
So we must understand what is the structure of RNA polymerase. This is what I have told you through Pathfinder. We would be looking at the animation also, understand the phases of transcription cycle, understand what is the difference between transcription and replication. So please do have a view here.
RNA polymerases are generally composed of several subunits. However, since RNA polymerase performs essentially the same vital function in all cells, it is not surprising that the RNA polymerases from all organisms share many features.
Majority of the RNA polymerases which are present in the organisms performs the same function. They have the function to biosynthesize the RNA molecule using the DNA as a template. So majority of the subunits which are being described in the prokaryotic RNA polymerase and the one which are present in eukaryotic RNA polymerase, they have a one-to-one correlation. They are exactly same with regard to each other.
Overall, RNA polymerase resembles a crab claw. A channel runs through the enzyme.
So overall, if you are going to look at the RNA polymerase, it resembles a crab claw-like structure. That means it resembles a crab-like structure which has a claw, and it is this claw through which the RNA chain is going to be biosynthesized. It would be constituted by the beta subunits, beta and beta dash, that will form the active site of the RNA polymerase enzyme. Next point.
Starting between the two pincers of the claw. The active site is found at the base of the pincers within a region called the active center cleft.
Now, there is a region which is present at the active site that is referred to as active center cleft. We would be looking at the structure as well as its analysis, that how it is going to biosynthesize this molecule. So please look here.
The active site contains two metal ions. This is consistent with the mechanism for nucleotide addition described for DNA polymerase. Like DNA,
Now, the active site contains two metal ions present within itself. The metal ions could be magnesium ion, manganese ion. These metal ions are going to inhibit the forces of repulsion in between the two phosphate molecules and are going to cause the attack of the oxyanion over the innermost phosphate. It will deplete a proton from the 3 prime oxygen OH molecule, and when the 3 prime OH molecule will get converted into an oxyanion, then the oxyanion is going to induce the attack to take place. So please do have a look here, that how the oxyanion is going to induce an attack. It will produce an oxyanion, and so see this, after the depletion, after the removal of this hydrogen which is going to take place, the oxyanion is going to be produced, and it will lead to the production of an oxyanion molecule. So this will lead to the production of oxyanion, and this oxyanion is going to attack the innermost phosphate. It is going to attack this innermost phosphate. Now, please do have a look that how it is going to attack.
DNA polymerase, RNA polymerase always adds nucleotides to the 3' end of a growing
See
polynucleotide.
this. The alpha phosphate has been removed, alpha phosphate has been joined, and the new nucleotide has been incorporated. The pyrophosphate, which is going to be produced here, is going to undergo degradation. This is the molecule of the pyroinorganic phosphate. Pyro inorganic phosphate, inorganic phosphate, PIP2. This pyroinorganic phosphate will undergo degradation, will lead to the formation of two molecules of inorganic phosphate, IP. Two molecules of inorganic phosphate will be produced, and this will produce very large amount of energy. This will produce large amount of energy, and this energy is used to drive the process of transcription polymerization. That this energy is used to drive transcription. How this energy is going to drive transcription? Let's have a review.
The transcription cycle is the series of events between binding of RNA polymerase to the target gene and dissociation of RNA polymerase and the completed RNA transcript from the DNA.
So the process of transcription could be categorized into three different parts. First process where the binding is going to take place and the first RNA transcript is going to be produced is referred to as initiation. When the RNA transcript is produced in its length, then the process is referred to as elongation. And ultimately, what is going to take place is termination when the RNA transcript is going to be terminated. Termination. So these are the three different processes which are going to take place: initiation, elongation, and termination. Now, please do have a look.
Transcription cycle is divided into three phases: initiation, elongation, and termination.
Okay, these are the three phases I have already told you.
Transcription initiation can be divided into three steps. In the first step, RNA polymerase binds to a region of DNA called a promoter. In bacteria, this step involves an initiation factor called sigma, which recognizes.
Now, the major subunits of the RNA polymerase have been described to you, and those subunits were alpha alpha dash, beta beta dash, and then there was an sigma factor which was present. Recently, another subunit named as omega has been discovered, whose function is still unknown. But this could be sigma RNA polymerase could be categorized into the following types. RNA polymerase can be of several different types. In the case of, when you are going to classify it in the case of prokaryotes, in case of prokaryote there is a single RNA polymerase. In case of prokaryotes, when you talk about, there is a single RNA polymerase. And this RNA polymerase which is produced here will have alpha alpha dash, beta beta dash, a sigma factor, and an omega. Okay. Now, this could be referred to as the core subunit. This is referred to as the core subunit. And another factor that is present is referred to as the sigma factor. Sigma factor. And here I'm writing alpha alpha dash, beta beta dash. Okay. While in the case of, when you talk about the eukaryotic cell, the eukaryotic cell has several different types of the RNA polymerase molecules. Now, which are the different types of the RNA polymerase molecules that are present in the case of a eukaryotic cell? In the case of a eukaryotic cell, this is all about prokaryotes. Then another one is all about eukaryotes. In the case of eukaryotes, there are three different types of RNA polymerases which are present. One is referred to as RNA polymerase 1, another one is referred to as RNA polymerase 2, and the third one would be referred to as RNA polymerase 3. RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. So those are the three different types of the RNA polymerase which are present in the case of eukaryotes. These are the three different types of RNA polymerases which are present in eukaryotes. So this is how the classification of the RNA polymerase is being made. Okay. Now moving forward.
Various sequences within promoters. The RNA polymerase with sigma attached binds the promoter in a defined orientation, so the same DNA strand is always transcribed from a given promoter. The RNA polymerase and promoter form a closed complex.
Now, RNA polymerase as well as the promoter region forms a very close complex with regard to each other because they are going to be closely associated. That is why they are forming a complex, and that type of complex is referred to as a closed complex. Now, the DNA is going to be melted. As the DNA is going to be melted, it will form an open complex.
In the second step, the closed complex undergoes a transition to the open complex.
Now, how the closed complex is going to undergo a transition into the open complex, that we will have to see that in what manner the open complex is being produced. Okay, so this is referred to as the first complex, that is referred to as the closed complex. When the RNA polymerase has been uploaded over the site of initiation, that is over the start point.
The pincers at the front of the enzyme clamp down tightly on the downstream DNA. Sigma also changes conformation, and the DNA strands separate, forming a bubble of single-stranded DNA.
It will lead to the formation of a bubble of single-stranded DNA molecule. Once a bubble of the single-stranded DNA molecule is being produced, the single-stranded DNA molecule is going to move forward. Okay, next.
In contrast with DNA polymerase, RNA polymerase is able to begin synthesis of a new polynucleotide without a primer. In bacteria, however, once RNA synthesis begins, the RNA polymerase goes through a period called abortive initiation. The enzyme synthesizes short RNA molecules of less than 10 nucleotides.
Now, the enzyme is going to synthesize very short RNA molecules, and those short RNA molecules are of less than 10 number of nucleotides present within the DNA. How it is going to biosynthesize, please do have a look. And
This is probably because
this is referred to as abortive initiation. Now, what is going to happen in the case of abortive initiation is that the RNA polymerase will form very short number of nucleotides of less than 10 base pair in length, so that the nucleotides which are going to be produced will be released outside, and then the process of transcription continues. This process takes place because the RNA exit chain over the RNA polymerase enzyme is actually being occupied by the sigma factor. Once the conformation of the sigma factor is altered, it leaves the RNA polymerase. The exit channel are open, and the RNA chain can effectively exit from the RNA polymerase. Do have a look.
a region of sigma partially blocks the RNA exit channel. Once this region has been ejected, and an RNA chain longer than 10 nucleotides has been synthesized, the elongation phase begins. Transition to the elongation phase is called promoter escape.
Now, this is important. Once the promoter escapes this abortive initiation or moves beyond the synthesis of those 10 number of nucleotides, and then they were again being aborted, then the process is commonly referred to as promoter escape. So now the promoter has escaped, and now the RNA polymerase is going to move forward.
During elongation, RNA polymerase unwinds the DNA in front of the enzyme, synthesizes RNA, proofreads RNA, dissociates RNA from the DNA, and reanneals the DNA behind the enzyme.
Now,
in contrast with DNA polymerase,
before we move towards the process of transcription, let us have a look at the process of initiation through Pathfinder. Okay, So just looking here at Pathfinder, this is RNA polymerase. Now, process of transcription, as I told you, the transcription can be divided into three stages: the initiation, elongation, as well as the termination. These are the three different stages in which the transcription could be effectively differentiated or divided. Now, the transcription begins when an RNA polymerase molecule binds to a promoter on the DNA sequence. Okay, the polymerase always moves in a particular direction. The RNA polymerase always moves in a 5 prime to 3 prime direction. The polymerase then moves along the template strand in the direction 5 prime to 3 prime by the step by step addition of ribonucleotide triphosphate until it reaches a signal that is referred to as termination signal, where the process of transcription is terminated. Now, initiation. What happens in the case of initiation? The sigma factor is going to recognize the first consensus sequences and is going to initiate the process through catalyzing the binding of the RNA polymerase to the promoter region. Initiation, it describes the synthesis of first nucleotide bonds in RNA molecule, how the first nucleotide bonds can be biosynthesized over the RNA molecule, the sequence of DNA needed for RNA polymerase to bind to the template and accompanies the initiation reaction, defines the promoter region. Okay, now which are the sequence which are being needed to initiate the process of transcription, defines by the promoter. A gene promoter can be defined as cis-acting DNA sequences necessarily required for the accurate and effective transcription of a particular gene. So, what is a promoter? Promoter is a cis-acting DNA sequence. Cis-acting means it is a position-dependent DNA sequence. Cis-acting means position-dependent. Position-dependent. What do you mean by cis-acting DNA sequence? Cis-acting DNA sequence means position-dependent DNA sequence, which is necessarily required for the accurate and effective transcription of a particular gene. It is necessarily required for the accurate and effective initiation of gene transcription. The DNA sequence of the promoter region is recognized by the requested RNA polymerase. Okay, so the DNA sequence is always being recognized by a specific enzyme, and that specific enzyme is referred to as the RNA polymerase. Now, this RNA polymerase recognizes certain specific DNA sequences, and those DNA sequences which are being recognized by the RNA polymerase are at the minus 35 region and at the minus 10 region, and those are the consensus sequences which are present. It consists of two 6 base pair consensus sequences. Minus 10 sequences referred to as the Pribno box. So this is the box that is present, and this box, which is the consensus sequences, is referred to as the Pribno box. This is Pribno box, which is present. Along with this Pribno box, there are other consensus sequences which are also present, okay, at the minus 35 region. The minus 10 sequence has the consensus sequence Tata, so it is also referred to as the Tata box, TATA, Tata box. Okay, now how the RNA polymerase is going to recognize where from the initiation has to take place. The RNA polymerase will recognize the site of initiation only after recognizing the specific sequences which are present over the Tata box. Only after it will bind to the Tata box and the consensus sequences. And these sequences are being recognized by a factor of RNA polymerase, and that factor is commonly referred to as the sigma factor. This is the factor that is going to recognize these consensus sequences. Okay, next. So every RNA polymerase will recognize certain specific sequences which are present. In the case of eukaryotes, there are several different types of sequences that are present. Okay. Next, moving forward. So, what do you mean by consensus sequences? Consensus sequences which are present here is a type of sequence that is being calculated by taking the different types of promoter sequences, comparing all the different types of promoter sequences so as to postulate which are the sequences that are going to be produced. So you can look here at the minus 35 region. There are several different types of consensus sequences that are present here, and at the minus 10 region also there are several different types of consensus sequences that are going to be present here. These refer to the different sequences that are present that will be transcribed. Okay. So consensus. So conserved, there are two terms. One was referred to as conserved sequences. Conserved sequences will never change. These will remain conserved, will remain same, will remain always same. For example, for example, telomere, telomere contain the sequence TTA GGG. This would be referred to as conserved sequence. Now, those sequences which are almost similar, but they can change. Sequences which are similar, sequences which are similar, sequences which are similar to each other, similar to each other, but has the ability to, but can change from one form to the other, and change from one form to the other are called consensus sequences. Such type of sequences are called as consensus sequences. Now the sigma subunit enables RNA polymerase to recognize specific site, and that site is referred to as the promoter site. In E. coli, promoter is going to interact with RNA polymerase, and this contains a unit that is referred to as sigma 70. Alternatively, several different types of sigma factors are present that recognize different consensus promoter sequences than sigma 70. There are several different types of sigma factors which are present that are going to recognize specific sequences. Sigma 54 is going to recognize this sequence. Sigma 38 is going to recognize this sequence. Sigma 32, this sequence. Sigma 28, this particular sequence. So these are several different types of sequences that are being recognized by several different types of sigma factors that are differs from each other and have different functions. Please do have a look. At least I must tell you that how the initiation takes place so that we can finish initiation. The holoenzyme promoter reaction starts by forming a closed binary complex. Firstly, there is a closed binary complex which is being produced. Closed means that the DNA is unwinded. Okay, DNA remains in the form of a double helical DNA molecule. Okay, the next step now, the transcription bubble is created by a local unwinding that begins at the site of the RNA polymerase. The next step that is being incorporated is the incorporation of the first two nucleotide, then a phosphodiester bond is being produced. This generates a ternary complex that contains RNA as well as a DNA and an enzyme. So there is a complex that is going to be produced, and that complex is called as the ternary complex. Now, how this ternary complex is going to be produced? The RNA, DNA, as well as the enzyme is present. You just look here. This is the sigma factor. This is the RNA polymerase, and this part is referred to as the sigma factor that is present. And it is this sigma factor to which which has the ability to recognize the specific sequences present over the DNA molecule, and the specific consensus sequences are present. There are specific SUS consensus sequences which are present. And once the specific consensus sequences will be present now, so those will be recognized by the sigma factor. Okay, once the sigma factor recognize those consensus sequences, it causes the melting of the dsDNA. This is dsDNA. The dsDNA has been melted. Oh, its melting state. Okay, now the melting that is going to take place, then the DNA is going to be melted, and this will be referred to as an open complex because the DNA has melted. So this is called as an open complex. Now the primer will be provided over the DNA that has been melted, and this will lead to the formation of the ternary complex. This would be called as the ternary complex. Ternary complex. This is called as the ternary complex, which is going to be produced. This is the ternary complex that is going to be produced because you have an RNA transcript. Once the sigma factor gets separated, then the sigma factor will be separated, and the RNA transcript is going to be produced in longer manner. This process is referred to as the elongation step. Okay, so please do have a look here. Ternary complex that contains RNA, DNA, and enzyme. This is the ternary complex that contains RNA molecule produced, DNA molecule, as well as the enzyme. Now, the process of abortive initiation is going to take place. Once abortive initiation is over, abortive initiation, I have just described the same in the animation. Now, there would be fragments of the RNA molecule being biosynthesized that would be expelled outside. Now, only the holoenzyme can initiate the transcription. Core enzyme has the ability to synthesize RNA on a DNA template but cannot initiate the transcription. RNA synthesis is frequently aborted, usually 2 to 3 times, up till 10 to 12 base pair of nucleotide has been joined. This phenomena that is going to take place is referred to be called as the abortive initiation. Once a polymerase manages to make a longer RNA chain of greater than 10 base pair, a stable ternary complex is being produced. This complex contains the enzyme, DNA template, and a growing RNA chain. So it will contain an enzyme, a DNA template, and a growing RNA chain that would be present. Okay, RNA chain is going to be biosynthesized, DNA template is going to be there, enzymes are going to be functional, and this process takes place in order that perfectly base paired or perfectly matched RNA template should be produced. RNA template should not be produced in a manner so that they should have low productivity, low reactivity. Only the RNA polymerase that will have the highest reactivity will be able to synthesize the RNA molecule. Why does abortive initiation takes place? Most important, abortive initiation takes place to maintain the efficacy of the transcription. Abortive initiation maintains the efficiency of transcription. It maintains the efficiency of transcription so that transcription should take place effectively only. Then only abortive initiation is going to take place. See this. Now, control of transcription initiation. There are two different types of genes which are going to be produced. Okay, before that, once a polymerase managed to make a longer RNA chain greater than 10 base pair, a stable ternary complex is being produced. The complex contains the enzyme, the DNA template, and the growing RNA chain present within itself. Now, in the case of bacteria, transcription initiation, when it is going to take place, when the transcription is going to be initiated, you will be able to find that there are two different types of controls. One is called as the constitutive control, another one is referred to as the regulatory control. What do you mean by constitutive control? What do you mean by regulatory control? Constitutive control depends upon the structure of the promoter. There could be two different types of promoter. One promoter could be a strong promoter and another would be referred to as a weak promoter. One could be referred to as a strong promoter. Strong promoter. Another would be referred to as a weak promoter. So these are the two different types of promoter regions which are present. Promoter regions have the ability to initiate the process of transcription. A strong promoter will strongly regulate the process of transcription. Weak promoter will weakly regulate the process of transcription. Promoter structures determine the basal level of transcription initiation. Promoter determine how the process of transcription is going to be initiated. The consensus sequence for the E. coli promoter are quite variable. Different promoters have different types of variable efficiencies. The efficiency is defined as the number of productive initiations that are being promoted per second. So, how many number of transcriptions are being produced per second is referred to as the productivity of the promoter region. That how many number of transcripts can be produced in the least amount of time. Okay, it may be a strong promoter or it can be a weak promoter. Strong promoters support high level of transcription initiation. So if you are going to talk about a strong promoter, it will produce very high amount of transcript as compared to the weak promoters. So let us look at the animation control and try to find out what are strong and weak promoters, as well as we should also look at the DNA polymerase and RNA polymerase. The major enzymes that are involved in the process of replication is called as the DNA polymerase. The major enzyme of the transcription is called as RNA polymerase. Now, DNA polymerase require deoxynucleotide triphosphate. RNA polymerase require ribonucleotide triphosphate. DNA polymerase has 10 subunit. RNA polymerase has 5. That is sigma, sigma dash, beta, beta dash, and then you have your sigma factor. Okay. So those are the different subunits which are present. Just a second. This would be beta and this would be beta dash. Beta dash. These are the 5 subunits. Now it is going to synthesize in 5 prime to 3 prime direction, 5 prime to 3 prime direction. Exonuclease activity. No exonuclease activity is present. Very important. But it has an exonuclease activity. The DNA polymerase one will have both the exonuclease activity, that is 3 prime to 5 prime, and 5 prime to 3 prime is also present. 5 prime to 3 prime is only present in DNA pol 1, which is also referred to as Kornberg polymerase. I have told you its structure as well as how it functions. Primer requirement, there is no primer requirement. Yes, there is a primer requirement. Configuration of the preferred DNA is duplex. Here it is single stranded. Metal ion requirement, yes, metal ion requirement is zinc and magnesium. Here also zinc and magnesium. Number of high energy groups consumed per nucleotide added is 2, and here it is also 2 number of nucleotides will be degraded. Now process of transcription. Now how does the process of transcription take place? I've already told you initiation, elongation, and termination are going to be the 3 different steps which are going to be involved. We have seen initiation. Now we would be looking at elongation. But before that, let us have a look at the animation. Now, we must understand how the unwinding of the DNA takes place. The major difference, helicase is being required here. In the case of DNA replication, the enzymes which are being required are DNA helicase. It requires DNA helicase. It requires primase. Neither helicase is being provided. No helicase is being provided. No primase is being provided. It requires single stranded DNA binding protein, also referred to as SSB proteins. But here, no SSB proteins are being required. Just clearing all the drawings and looking at the animation.
RNA polymerase is able to perform these functions without the assistance of other proteins.
So RNA polymerase has the ability to catalyze the entire process without the assistance of any other protein that could be involved. Next point.
The proofreading activity of RNA polymerase is less efficient than that of DNA polymerase. While the proofreading activity of DNA polymerase only allows about one mistake per 10 million nucleotides, the proofreading activity of RNA polymerase allows about one mistake per 10,000 nucleotides.
Okay. Now, both the DNA polymerase and RNA polymerase have different types of proofreading activity. I've already told you that the proofreading activity, previously it was being thought that the process of transcription do not have any proofreading activity, but now it has been confirmed that yes, it has a very well proven proofreading activity present within itself. Previously, before 2005, it was previously before 2005 it was thought that RNA biosynthesis synthesis or transcription lacks any proofreading activity. This is important. Changing the color. Transcription lacks any proof reading activity. But now it has been illustrated that two different types of proof reading activity are present. These includes the following different types. One is referred to as hydrolytic proof reading activity, and another one is referred to as phosphorolytic proof reading activity. Okay, so these are the two different types of proofreading activity which are present. One is hydrolytic proofreading activity, and another one would be referred to as phosphorylated proofreading activity. Now, how these activities takes place, that we have to see. Okay.
RNA polymerase has two proofreading mechanisms. The first mechanism is called pyrophosphorylated editing and involves a simple back reaction to
Now, the first mechanism is called as pyrophosphorylated editing, in which there is a simple back reaction that is going to take place. Once the simple back reaction that is going to take place, then it is going to be moved back. And once it moves back, it is going to hydrolyze the molecules that hydrolyze the RNA template molecules that have been joined to the DNA, and then it is going to be released out.
catalyze the removal of an incorrectly inserted ribonucleotide. RNA polymerase slows down when an incorrect ribonucleotide is added, making removal of an incorrect base more likely than removal of a correct base.
So it will cause the removal of an incorrect base, whatever is present, rather than removal of a correct base molecule. So there would be an incorrect base that could be effectively removed. Next point.
The second mechanism is called hydrolytic editing and involves backtracking of the polymerase by one or more nucleotides to cleave the RNA product.
Now, what do you mean by backtracking? Backtracking is another method that could be used. In the case of backtracking, the RNA polymerase could move back. Now, it was moving in one direction, it moves back, uses a factor called as Grey factor, and then dissociate the incorrectly associated nucleotides.
In bacteria, hydrolytic editing is stimulated by GRE factors, which also serve as elongation stimulating factors.
Now, there are specific factors which are present. Those are referred to as the Grey factor, also referred to as the elongation stimulating factor, that will be stimulating the process of elongation to take place so that the DNA molecule, so that the RNA chain can be elongated further.
Sequences called terminators trigger the elongating polymerase to dissociate from the DNA and release the completed RNA chain. This animation describes bacterial transcription termination.
Now, the other sequences that are going to be functional are referred to as the terminating sequences. But before we move towards the terminating sequences, we must know about elongation. So, looking at elongation from the Pathfinder series. So, look here, elongation involves the movement of the transcription bubble by a disruption of the DNA molecule. The DNA molecule is going to be disrupted and the elongation is going to proceed. The enzyme that moves along the DNA and extends the growing RNA chain is referred to as the RNA polymerase. So, RNA polymerase is the enzyme that is going to move and is going to elongate and biosynthesize the RNA molecule. As the enzyme moves, it unwinds the DNA molecule at its front and exposes a new segment of the template to be biosynthesized. The RNA DNA hybrids are going to be produced. Ultimately, during each nucleotide addition, the beta and gamma phosphates are being released. As the beta and the gamma phosphates are going to be released from the incoming nucleotide, the hydroxyl group is going to be removed. The overall reaction rate is 40 nucleotides per second at 37 degrees Celsius. This is the rate of replication that is going to take place. The RNA, as RNA polymerase is going to move forward along with the DNA duplex, it generates positive supercoil ahead and leaves negative supercoil behind it. So RNA polymerase is going to move forward. It generates positive supercoiling ahead and leaves a negative supercoiling at its back, because the DNA which is present in us is negatively supercoiled. That is why sir is going to write in such a manner that sir is considering the DNA to be that of human beings. Okay, so when the DNA, which is DNA, is negatively supercoiled in humans, DNA in humans and eukaryotes, DNA is negatively supercoiled. DNA is negatively supercoiled. It's negatively supercoiled. Supercoiled. So it has to be relaxed. So it has to be relaxed. So it is relaxed by introducing positive supercoiling. By introducing positive supercoiling. Positive supercoiling. Supercoiling ahead and leaving negative supercoiling behind. The enzyme that is going to participate here is topoisomerase 1, and this will be referred to as the DNA gyrase. Okay, the inhibitor that is present is referred to as the cordycepin. Cordycepin is the inhibitor that is going to inhibit the process of elongation to take place. In addition to the 3' end of the RNA chain further elongates. RNA is going to be further elongated, and it is the inhibitor for this particular reaction is cordycepin. Now, what do you mean by transcription termination? There are two different types of transcription termination. One is referred to as row dependent, another one is referred to as row independent. So let us look at what is row dependent and what is intrinsic termination, what is extrinsic termination. Uh, the lecture will be a bit long. Please have some patience. We are going to finish this topic very soon. So kindly look at the termination process.
Bacteria have two types of terminators, row independent and row dependent.
Now, there are two different types of terminator sequences which are present in bacteria. One is called as row independent, another one is referred to as the row dependent terminator sequences. Now, what do you mean by row independent and row dependent terminator sequences? Row independent terminator sequences will be referred to as the intrinsic termination, and this will require another protein. Row dependent will require another protein called as the row protein.
Row-independent terminators, also called intrinsic terminators, consist of two sequence elements, a short inverted repeat of about 20 nucleotides and a stretch of about 8 AT base pairs.
So an inverted repeat of 20 nucleotide is present and a stretch of 8 base pair is present. This is an inverted repeat. Then there is a stretch of 8 base pair. Okay. Now, this inverted repeat will cause the formation of an inverted loop shaped structure. How?
The RNA that results from the inverted repeat sequence is able to base pair with itself and forms a hairpin structure. The hair
Now see this. This is an inverted repeat. They this is a loop region. This is a gap region present in between the inverted repeats. The inverted repeats will be able to base pair with each other and forms a helical loop shaped structure.
hairpin then disrupts the elongation complex.
Now see this. This is a hairpin loop-like structure that is going to be produced. The hairpin will disrupt the elongation concept complex. The hairpin then disrupt the elongation complex. Hairpin will not be able to will not be able to process through the process of elongation. AU base pairs are the weakest of all base pair, allowing the RNA to easily dissociate from the DNA molecule. Look here.
AU base pairs are the weakest of all base pairs, allowing the RNA to easily dissociate from the DNA.
This is how RNA can be effectively dissociated from the DNA molecule. Now the RNA can be effectively dissociated, so the RNA molecule has been released out.
Rho-dependent terminators are less well characterized, but optimally consist of stretches of about 40 nucleotides that do not fold into a secondary structure in the RNA transcript. Rho-dependent terminators require the action of the Rho factor, a ring-shaped hexameric
Now, row determining transcription termination requires a factor that is referred to as the row factor. Now, row factor is a type of factor that will bind to the mRNA molecule and will terminate the process of transcription. How it is going to terminate the process of transcription, that we have to see. Please have a look.
protein. The Rho factor binds to the transcribed RNA as it exits the polymerase. Using the energy derived from ATP hydrolysis, the Rho factor pulls the RNA from the template and RNA polymerase.
Now, row factor is going to bind to the mRNA transcript. It will be able to pull the mRNA transcript away in such a manner so that the mRNA transcript is being expelled off. The RNA polymerase is going to move away from the effective sites of the transcription. So this is how row dependent termination is going to take place.
Since RNA polymerase dissociates the RNA transcript from the DNA as it is transcribed, multiple RNA polymerases can transcribe the same gene at the same time. This
Now, there are many number of RNA polymerases that can transcribe the same gene at the same time. So this process is referred to as RNA polymerase cycling. This is referred to as RNA polymerase cycling that is going to take place. One RNA polymerase is going to precede another RNA polymerase is going to precede another. So the RNA polymerase are going to move one behind the other, and this process is called as ribosomal RNA polymerase cycling.
allows a cell to synthesize a large number of RNA transcripts from a single gene in a short time.
Okay, this allows the cell to synthesize very large number of RNA transcript from a single gene. So there are large number of RNA transcript which are going to be produced from a single gene. Next.
How well do you understand transcription? In this section, you will find out.
Now, before we move to the application based section, let us look at the process of transcription and finish this off through Pathfinder. So I have already told you that there are two different types of termination processes. So exactly how termination is going to take place, that is not known, but bacteria seems to have two different types of termination mechanisms. One is called as the intrinsic termination mechanism. Another one is called as the row dependent termination mechanism. Now, what do you mean by intrinsic termination mechanism? What do you mean by row dependent termination mechanism? That we have to look here. Now, intrinsic termination has been thought to promote dissociation of the polymerase by destabilizing the attachment of the growing transcript to the template. Now, intrinsic termination is the first type of termination that is going to take place. Now, how intrinsic termination takes place, so that we have to see. Intrinsic termination has been thought to promote dissociation of the polymerase by destabilizing the attachment of the growing transcript to the template. The growing transcript is going to be destabilized. As it is going to be destabilized, so the transcription termination will occur. Intrinsic termination consists of formation of a hairpin loop like structure in the RNA product followed by U-rich region in which termination occurs. The typical distance between the hairpin and the U-rich region is referred to as 7 to 9 base pair. The typical distance between the hairpin loop-like structure and the U-rich region is just 7 to a 9 number of base pair. Here it will form a loop-shaped structure that you can find because of the inverted repeat just coming in this section. Because of the inverted repeat, this is referred to as the termination site. Termination site, and this is the termination site where from the process of where the process of termination is going to take place. Okay, here the process of termination is going to take place, and this termination site has two different types of inverted repeat sequences. These inverted repeat sequences can base pair with each other. This is one inverted repeat sequence, and there is the other inverted repeat sequence. These two inverted repeat sequences have undergone base pairing with regard to each other. As the two inverted repeat sequences have undergone base pairing, so what is going to happen? They have formed complementary bases with regard to each other. As the two have formed complementary bases, there is a loop shaped region which is present. Here you can find this is the loop shaped region which is present. Okay, and now it is going to perform the process of termination. How? Please have a look. This will cause the RNA polymerase that would be present here in this manner to halt. RNA polymerase will halt. RNA polymerase will halt here. When RNA polymerase is going to halt, it is going to be stopped. Next point. The next point is the second type of termination is row dependent. Row protein is actually an ATP dependent RNA polymerase helicase and has the ability to disrupt the RNA DNA complex. It is a 275 kilodalton protein. It binds to RNA, translocates along the RNA until it reaches RNA DNA hybrid, and then ultimately it is going to release the RNA from the DNA template. So this is how row is going to be functional. Okay, now, looking at certain questions from the transcription that has been asked in CSIR and other net entrance examination, what is the minimum length of the time required for the synthesis of E. coli polymerase on an average encoding 100 kilodalton protein? So, if you are going to talk about 100 kilodalton protein, so 100 kilodalton protein will have 910 residue. How can you say that? Weight of each residue, weight of each residue is equal to 110 Dalton. Okay. So 100 kilodalton protein. 100 kilodalton means 100 into 1000 Dalton protein. Into 1000 Dalton protein. This is protein. Okay. This protein is being divided by the weight of a single amino acid. Dalton will give you number of amino acid. So this is the number of amino acid calculated. Okay. Now, each amino acid is being encoded by a codon. Each amino acid encoded by a codon. By a codon. So, number of amino acid 910 into 3 will give you number of nucleotides equal to nucleotides, and that is equals to 2730 number of nucleotides. Okay, at a maximum transcription rate of 50 nucleotide per second, at a maximum transcription rate of 50 nucleotide per second. The proteins will be synthesized at a speed of 56.6 seconds. Why is RNA synthesis not a carefully monitored as compared to DNA synthesis? Because see, in the case of DNA synthesis, there are several different enzymes which are going to take place. Every DNA polymerase is different. In the case of RNA synthesis, the transcription, there is a single RNA molecule that is transcribing the entire process. It is working as a helicase, it is working as a primase. There, in the case of DNA replication, there are several separate enzymes. So the DNA replication has a much high processivity, has a high accuracy as compared to RNA synthesis. An error will only affect one molecule of the mRNA of many synthesized from a gene. In addition, the error do not becomes a permanent part of the genomic information. The error will not become a permanent part of the genomic information. Di and trinucleotides are occasionally released from the RNA polymerase at the very start of the transcription, a process called as abortive initiation. What do you mean by abortive initiation, and how does the process of abortive initiation takes place? The process restart at the process of transcription. Suggest a plausible explanation for abortive initiation. The base pairing energy of all the di and the trinucleotides differs from each other at the beginning of transcription, is not sufficient to prevent strand separation and loss of products. Okay, so what has to be done? So there is a process that is going to take place, abortive initiation, because the energy which is present in between the di and the trinucleotide is at the beginning of transcription is not sufficient to prevent strand separation and loss of product. Because you look here na, at the very beginning of transcription, the energy that is present is very low. Because the energy is very low, that is why the process of transcription cannot take place, as the energy of the transcription is very लेस, क्योंकि ट्रांसक्रिप्शन की एनर्जी बहुत कम होती है, इसलिए अबॉर्टिव इनिशिएशन जरूरी है. दैट इज व्हाई अबॉर्टिव इनिशिएशन is needed, so that effective binding of the RNA to the DNA is going to take place. Then only the process of transcription begins, and it should have higher accuracy. Okay, now that's all. We have finished the process of transcription. Let us try to solve some of the questions.
Initiation. Help this bacterial RNA polymerase bind the DNA and begin transcription.
Now, the bacterial uh, transcription is going to begin.
Question 1. Which
Transcription
factor
is
is
going to start.
required for this step of transcription?
Now, first step that is being required is the binding of the sigma
Correct.
factor. The sigma factor will bind to the RNA polymerase first, and then see this.
Question 2. Where does the RNA polymerase with sigma attached bind to the DNA? Be sure to match the orientation of the gene.
Now, where it is going to bind? It is going to bind to a specific site which has the gene orientation. It is going to bind here.
Correct.
Okay. In the direction of
Question 3. Which component or components of the closed complex must change conformation during transition to an open complex?
Now, all the components should always change conformation for the transcription to take place, because the DNA has to be melted. The double-stranded DNA has to be converted into single-stranded DNA. It should change conformation. For the process of effective transcription, the sigma factor must leave the RNA polymerase. So it should change conformation. The RNA polymerase should also alter its conformation.
Question 4. Once the open complex has formed, the RNA polymerase goes through a period called abortive initiation. Which component of the open complex must change conformation before promoter escape?
Now, how promoter escape should take place? Promoter escape should take place only when the sigma factor will change its orientation. Once the sigma factor changes its orientation, promoter escape is going to take place. Next.
Elongation. Help this bacterial cell form a functional RNA transcript. Question 5. Which enzyme or enzymes are responsible for these activities during elongation?
Now, which of the following enzymes will be responsible for the different activities during elongation? When you talk about, then how to answer? First, I cannot place any of these. Where can I place? Only grief factor is given. Okay, I can tick them. So I should move forward. What should I do? Unwinding the DNA in front of the enzyme. Okay. So first, RNA polymerase.
Question 6. Match the name of this proofreading mechanism with the protein that stimulates this activity.
So it is hydrolytic editing, and the factor is grief factor.
Correct. Termination. Help this bacterial RNA polymerase dissociate from the DNA. Question 7. What mechanism of termination does this terminator most likely utilize?
Now, because the distance between the two terminating sequences must be equal to 7 to 8 number of base pair, and it is less than 7 to 8 number of base pair, so it will utilize row dependent termination. If the distance would have been 7 to 8 number of base pair, it would have effectively used the intrinsic or the row independent termination sequences. It is all about the distance which is present in between the inverted sequences. The distance between the inverted sequences is less than 8 base pairs, so it requires row dependent termination.
Correct. Question 8. Which protein is necessary for this mechanism of termination?
Now, the protein that is responsible is referred
Correct.
to the rho protein. So, rho protein is going to bind, it is going to dissociate the entire process.
The primary enzyme of transcription is RNA polymerase. Both prokaryotic and eukaryotic RNA polymerases resemble a crab claw.
Both of these resembles a crab claw. Just the conclusion of the
RNA polymerases are generally composed of several subunits.
alpha alpha dash, beta beta dash, and sigma, and then your omega subunit. Sigma subunit is never is referred to as a different factor. Others are referred to as core subunits.
The active site of RNA polymerase contains two metal ions. Just as in DNA polymerization, these metal ions promote the addition of an incoming nucleotide to the 3' end of the growing RNA chain.
Okay, next point.
The series of events between binding of RNA polymerase to the target gene and dissociation of RNA polymerase and the completed RNA transcript from the DNA is called the transcription cycle. The transcription cycle is divided into 3 phases: initiation, elongation, and termination.
Now, this is the 3 different phases which are present. One is initiation, another one is elongation, and the third is termination.
Transcription initiation can be divided into 3 steps: binding of RNA polymerase to form a closed complex, transition to an open complex, and promoter escape. In bacteria, an initiation factor called sigma recognizes various sequences within promoters.
So there is a factor sigma that recognizes all these sequences. And
during elongation, RNA polymerase unwinds the DNA in front of the enzyme, synthesizes RNA, proofreads RNA, dissociates RNA from the DNA, and reanneals the DNA behind the enzyme.
So this is all the function that RNA polymerase is going to function. It is going to synthesize the RNA molecule. It is going to proofread the RNA molecule. It is going to dissociate the RNA molecule, reanneal the DNA behind it, and then the process of termination occurs.
During termination, sequences called terminators trigger the elongating polymerase to dissociate from the DNA and release the completed RNA chain. Bacteria have 2 types of terminators: row independent and row dependent.
See, this is row independent, this is row dependent termination, and this is how termination is going to take place. They are going to be released.
You have completed this exercise.
That's all. this is a very important chart. You can note this particular chart. That's all for today. We have completed the chapter. We have completed the process of transcription. If you feel any doubt, you just type here and I will try to answer all your queries. Thank you. Thanks a lot. Good night. Goodbye.